Deoxyribonucleic acid, or DNA, is the blueprint of life. It contains the instructions for building and maintaining an organism. This intricate molecule holds the key to understanding our inheritance, our health, and our very existence. DNA's complexity is captivating. The sheer volume of information encoded within this microscopic structure is astounding. It dictates everything from the color of our eyes to our susceptibility to certain diseases. DNA is a remarkable feat of biological engineering. Its structure is both elegant and functional, allowing it to store vast amounts of genetic information in a compact form. The study of DNA has revolutionized our understanding of biology and medicine. It has led to groundbreaking advancements in fields such as genetic engineering, disease diagnosis, and personalized medicine. Unraveling the secrets of DNA has been one of the greatest scientific achievements of our time. It has opened up new avenues for scientific exploration and medical innovation. As we delve deeper into the world of DNA, we continue to uncover new insights into the fundamental mechanisms of life. The journey into the world of DNA is an exploration into the very essence of our being. It is a testament to the power of scientific inquiry and the boundless potential of human ingenuity. DNA is primarily located within the nucleus of a cell, a specialized compartment that serves as the control center of cellular activity. This nuclear DNA, often referred to as chromosomal DNA, is organized into long, linear structures called chromosomes. Each chromosome carries a unique set of genes, which are segments of DNA that code for specific traits. However, DNA is not exclusive to the nucleus. Some DNA is found outside the nucleus, in organelles called mitochondria. These organelles are responsible for cellular respiration, the process by which cells convert nutrients into energy. Mitochondrial DNA, or mtDNA, is distinct from nuclear DNA and has its own unique inheritance pattern. The presence of DNA in both the nucleus and mitochondria highlights the intricate organization of eukaryotic cells. The compartmentalization of DNA allows for efficient regulation of gene expression and cellular processes. The existence of extranuclear DNA, such as mtDNA, further enriches the genetic landscape of an organism. Understanding the location and types of DNA is crucial for comprehending the flow of genetic information within a cell. It provides insights into the mechanisms of inheritance and the interplay between nuclear and extranuclear genomes. The structure of DNA is perhaps its most iconic feature. DNA exists as a double helix, resembling a twisted ladder. This elegant design is not merely aesthetically pleasing, but also functionally significant. The double helix structure provides stability and facilitates the storage and transmission of genetic information. The two strands of the DNA double helix are composed of alternating sugar and phosphate molecules, forming the backbone of the structure. Attached to each sugar molecule is one of four nitrogenous bases: adenine A, thymine T, guanine G, and cytosine C. These bases are the informational units of DNA, much like letters in an alphabet. The two strands of the DNA double helix are held together by hydrogen bonds between the nitrogenous bases. The specific pairing of these bases, A with T and G with C, is known as complementary base pairing. This precise pairing ensures the fidelity of DNA replication and the accurate transmission of genetic information from one generation to the next. The double helix structure of DNA is a testament to the remarkable elegance and efficiency of nature's design. It is a testament to the power of simple building blocks, arranged in a precise manner to create a molecule capable of storing and transmitting the blueprint of life. Nucleotides are the fundamental building blocks of DNA. Each nucleotide consists of three components: a sugar molecule, a phosphate group, and a nitrogenous base. The sugar molecule in DNA is deoxyribose, giving DNA its full name, deoxyribonucleic acid. The phosphate group links the sugar molecules together, forming the backbone of the DNA strand. The nitrogenous base, attached to the sugar molecule, carries the genetic information. The sequence of these bases along the DNA strand determines the specific instructions encoded within the DNA molecule. There are four types of nitrogenous bases in DNA: adenine, A, thymine, T, guanine, G, and cytosine, C. These bases are categorized into two groups: purines, A and G, and pyrimidines, T and C. The specific arrangement of these bases along the DNA strand constitutes the genetic code. Nucleotides are the essential units of information storage in DNA. Their precise arrangement dictates the production of proteins, the workhorses of the cell, and ultimately determines the traits and characteristics of an organism. The two strands of the DNA double helix are held together by hydrogen bonds between the nitrogenous bases. This bonding is not random. Each base has a specific pairing partner. Adenine, or A, always pairs with thymine, known as T, and guanine, or G, always pairs with cytosine, C. This precise pairing is known as complementary base pairing. Complementary base pairing is crucial for the stability and functionality of DNA. The hydrogen bonds between the base pairs provide the force that holds the two strands of the double helix together. This pairing also ensures the fidelity of DNA replication, as each strand serves as a template for the synthesis of a new complementary strand. The specific pairing of bases also has implications for the genetic code. The sequence of bases along a DNA strand determines the sequence of amino acids in a protein. Each three-base sequence, called a codon, codes for a specific amino acid. Complementary base pairing is a fundamental principle of DNA structure and function. It ensures the accurate transmission of genetic information and underpins the mechanisms of inheritance. The sequence of nitrogenous bases along a DNA strand constitutes the genetic code. This code dictates the production of proteins, which are responsible for a vast array of cellular functions. Proteins are made up of amino acids, and the sequence of bases in DNA determines the sequence of amino acids in a protein. Each three-base sequence in DNA, called a codon, codes for a specific amino acid. There are 64 possible codons, but only 20 amino acids are used to build proteins. This redundancy in the genetic code allows for some flexibility and error tolerance. The genetic code is universal, meaning that the same codons code for the same amino acids in all organisms. This universality is a testament to the shared evolutionary history of life on Earth. It allows for the transfer of genetic information between different species, as demonstrated by genetic engineering techniques. The genetic code is the language of life, a set of instructions that dictates the production of proteins and ultimately determines the characteristics of an organism. Understanding the genetic code is fundamental to comprehending the mechanisms of inheritance and the diversity of life. Mitochondria, the powerhouses of the cell, possess their own DNA, distinct from the nuclear DNA found in chromosomes. Mitochondrial DNA, or mtDNA, is a circular molecule that carries a limited set of genes, primarily involved in the process of cellular respiration. mtDNA has its own unique inheritance pattern. Unlike nuclear DNA, which is inherited from both parents, mtDNA is inherited maternally. This means that an individual's mtDNA is identical to their mother's mtDNA. The maternal inheritance of mtDNA allows for the tracing of lineages through the maternal line. It has been used to study human evolution, population migrations, and historical relationships between different groups of people. mtDNA is a valuable tool for understanding human history and the evolutionary relationships between different populations. Its unique inheritance pattern provides insights into the maternal lineage and the genetic diversity of human populations. The study of DNA has revolutionized our understanding of biology and medicine. It has led to groundbreaking advancements in fields such as genetic engineering, disease diagnosis, and personalized medicine. DNA technology has become an indispensable tool in various scientific disciplines and medical practices. Genetic engineering allows for the manipulation of DNA sequences, enabling scientists to modify the genetic makeup of organisms. This technology has applications in agriculture, where it is used to develop crops with enhanced traits, and in medicine, where it holds promise for gene therapy and the treatment of genetic diseases. DNA sequencing, the process of determining the order of bases in a DNA molecule, has become increasingly rapid and affordable. This technology has revolutionized disease diagnosis, enabling the identification of genetic mutations associated with various diseases. It has also paved the way for personalized medicine, tailoring treatments to an individual's genetic makeup. DNA technology has transformative potential in various fields, from agriculture to medicine. It has opened up new avenues for scientific exploration and medical innovation, promising a future where our understanding of DNA translates into tangible benefits for human health and well-being. DNA, the blueprint of life, continues to fascinate and inspire scientists. Our understanding of DNA has grown exponentially in recent decades, yet much remains to be discovered. The future of DNA research holds immense promise for unraveling the complexities of life and developing innovative solutions to global challenges. One of the most exciting frontiers of DNA research is the study of epigenetics, the mechanisms by which gene expression is regulated without altering the underlying DNA sequence. Epigenetic modifications play a crucial role in development, aging, and disease. Understanding these modifications could lead to new therapies for a wide range of conditions. Another promising area of research is the development of gene editing technologies, such as CRISPR-Cas9, which allow for precise modifications of DNA sequences. Gene editing has the potential to revolutionize medicine, offering cures for genetic diseases and novel approaches to cancer treatment. The journey into the world of DNA is an ongoing exploration into the very essence of our being. As we continue to unravel the secrets of DNA, we gain a deeper understanding of life's complexities and unlock new possibilities for improving human health and well-being.